Good morning, everyone. Welcome to session 5 of the Middle East Symposium for HCA. It's a great pleasure to be here with you this morning. My name is Lucia Robson, and I'm a Senior Program Manager working in the HCA Executive Office. I'm moderating a really exciting session this morning. I'm particularly interested in the 3 topics that we have. I'll introduce the 3 speakers, and then we can start off. So first, we have Fereshteh Fallah, who is a PhD student at Tehran University, who's speaking to us about the role of the tumor microenvironment communication in cancer progression. Then we have Dr. Tala Shahin, who is a postdoctoral associate at NYU Abu Dhabi, who has an interesting talk on how single-cell genomics can help in the response to malaria, especially in children in West Africa. And finally, Priyam Narain, who is a researcher interested in circadian rhythms and mood disorders, particularly in the mouse brain. As with all the sessions, I really encourage anyone to ask questions in the Q&A. If we don't get time for them, the speakers will hopefully follow up afterwards. So encourage you to ask as many questions as you'd like. Without further ado, Fereshteh, I'll hand it over to you. Please present your slides and begin when you're ready.
Okay, do you have my slides and my voice clearly?
Very well, thank you.
Okay. Hi, it's an honor to be here at the first Human Cell Atlas Middle East Symposium. I am Fereshteh Fallah from Tehran University, and I'm excited to share with you my recent project. The title of my today's talk is The Role of Tumor Microenvironment Communication in glioma cancer progression using single-cell RNA sequencing. I conducted this research under the supervision of Dr. Kaveh Kawusi from Tehran University and Dr. Laleh Haqverdi from MDC. And in the introduction, I will briefly speak about tumor microenvironment, single-cell RNA sequencing, and current method to investigate the TME by single-cell RNA sequencing. Let's start. Okay, to embark on this scientific journey, it's essential to unravel the complexities of tumor microenvironment. These dynamic ecosystems, known as TME, are the heart of cancer progression. I should present a definition for it. TME is an ecosystem comprising cancer cell, infiltrating immune cell, and diverse cell type that coexist with each other. A wide variety of mechanisms are involved in the communication between malignant and surrounding cell, which can ultimately result in fast proliferation, metastatic, tumor resistance, or can act as tumor suppressor. Now, imagine having a magnifying glass for molecular world of TME. This is where single-cell RNA sequencing comes into play. It's like reading the individual stories of each cell, decoding their gene expression, and understanding the intricate orchestration of cancer symphony that perform between cancer cell and other cells in TMEs. The study of cell-to-cell communications and crosstalk between tumor cell and other cell types, such as immune cell and in its microenvironment, will enable new frontiers for effective cancer treatment. In other words, to deeply investigate TME, we require a model that can quantify both how signals are transferred between cells and how that information is processed within cell. As you know, various methods exist for extracting this interaction, including cell phone DB, single cell R, and etc. But these tools only consider the ligand receptor interaction and the regulatory network involving transcription factors and targeted genes. It didn't consider in this model. In light of this important factor, the purpose of our pipeline, the purpose of our proposed pipeline for exploring molecular mechanisms in 2 stages of glioma cancer, grade 3 and grade 4, by integrating, a single-cell RNA sequencing data and constructing a multi-layer network that capture multiple layers of interaction, such as ligand-receptor pairs across senders and receiver cells, and also gene regulatory network in the receiver cells. In this figure that I presented for you, I want to illustrate the presented pipeline that's enabling us to examine how communication between immune cell and cancer cell evolve in different glioma stage and the multi-layer network behind this interaction, as you can see at the final part of this figure. In the method and material method and results section, I want to explain what data selected, or which data selected, what we did in pre-processing part, how we did downstream analysis, a multi-layer signaling network creation pipeline, and finally quartile-based prediction model, and what we extracted and in which way we distracted a quartile module. We use publicly available data that consists of 6,341 cells from 6 patients, 2 for grade 3 and 4 for grade 4 glioma. In this figure, you can see the resulted quality control part from Surat pipeline. This part should be done in order to be sure that only high quality cell proceed to the following steps. In the figure, you can examine the statistics of analysis sample. In part B, you can see the detected gene profile per sample, total gene count per sample, and other statistics that you can extract from this pipeline. After quality control and drawing away cells with low quality, a computational pipeline started in my work. We have a normalization matrix at the start point of our study, and then same analysis was done for grade 3 and grade 4 of my samples. We did feature selections. After that, PC selection or dimension reduction. After that, we use UMAP for visualization, and all of these steps did in the Surat package. And as you can see, 3 main cell types or 3 main clusters detected. And after gene marker identifications part, we can see cell type annotation that we did in our study. And we found 3 cell types: cancer cell, immune cell, and OPC cells, or normal cell for this cancer type. I hope you can see the figure because all of text is tiny. I apologize for that. Let's continue. After cell annotation part and finding different cell types that exist in our data, we can start constructing multi-layer network. This network connect glioma cells or cancer cell as a receiver cell and immune cell as sender cell. This multi-layer signaling network, as you can see in the figure, consists of 4 layers: ligand layer, receptor layer, TF or transcription factor layer, and targeted gene layer. For creating edges in each layer, we use some databases and statistical sets such as Fisher test. As you can see in each layer, by moving from grade 3 to grade 4 glioma, the number of interaction decreases. And this occurred as a result of cancer cell desire to be more independent and their dislike of receiving connection from other cell types when they go to the metastatic stage. We also examine the topology. The topological difference between grade 3 and grade 4 multi-layer network. As shown in this figure in the top row, the node degree and the number of age, in the grade 3 network was significantly larger than on the grade 4 network. And for the part below figure in the bottom, you can see how different centrality measurements and criteria have changed over stages. You can see Katsa centrality, PageRank centrality, and betweenness centrality between nodes in two stages that are significantly different with each other. These results evidently show that some nodes in one network were highly important, but they nodes don't play a significant role in another stage. For example, by considering degree centrality, the NYC gene is a crucial transcription factor in grade 4, but these genes didn't find in the grade 3. And these genes, according to previous study, play a crucial role in regulating the cell cycle and metabolism. NYC is a compelling therapeutic target in grade 4, but despite considerable attempt, direct inhibition of NYC-TF was remained a challenge. Okay, as all pipelines suggested, indirect strategies should be used for inhibiting NYC genes. For example, inhibit the ligand receptor interaction or inhibit ligand releasing in the immune cell. Besides this gene that I presented for you, we found many other important connections and listed them as potential therapeutic agent for further examination. After topological analysis, we should do functional analysis too. In order to decode the secret hidden within the data, we extracted biologically meaningful quadrupel using reactome-fizz tools. What I meant by quadrupel is a sequence of ligand, receptor, transcription factor, and target genes. In grade, in grade 3, we identified 2,290 such cascade, and in grade 4, the number is 349. And so these quadrupel can be act as all feature sets for machine learning tasks. So instead of using all genes or just differentially expressed gene between two stages, we use each quadrupel separately as feature for all machine learning tasks.
Fereshteh, this is your 1 minute. 1 minute.
Okay. In the machine learning part of our study, we developed a predictive model to understand the transition from grade 3 to grade 5. And by some criteria among these quadrupel, one was found to effectively differentiate between two classes, and it was a quadrupel consists of PDGFR as ligand, PDGFR A as a corresponding receptor, CREB1 as its transcription factor, and finally PLAT as its target gene. We evaluated the performance of our model with selected quadrupel using 100 iteration of a 5-fold cross-validation test. And in order to ensure that we didn't input any bias to our analysis, the data set were shuffled with different random seeds before splitting in each cross-validation iteration. And this model performed in different classification algorithms such as SVM and random forest web, and we get 87% F1 score and accuracy among all different machine learning algorithms. But originally, doesn't stop at the prediction, and we looked beyond the transition and examined the association between selected quadrupole and patient prognosis. This model demonstrated high prognosis accuracy, as you can see in the figure part B, and we also examined the power of our quadrupole model with another biomarker that established, and the model accuracy in predicting overall survival in glioma patients suppressed the other marker that reported in another papers. Okay, thanks. I can't move to discussion part. Thanks for your time and your attention. I welcome any questions.
Fereshteh, thank you so much for your presentation. Really interesting, really clear. I encourage anyone to ask any questions in the chat. And we can answer them now. Otherwise, Fereshteh can answer them in the course of the session. So it looks like there's no Q&A at the moment. So Fereshteh, thank you so much.
Thank you.
I'd now like to present Dr. Tala Shahin from NYU Abu Dhabi. And Tala, when you're ready, please take it away.
Yeah. Okay, just sharing my slides. Okay, so you can see my slides, I guess, and me.
Very well.
Okay, perfect. So thank you, Lucia, for the introduction. And hello, everyone. So I'm Tala, a postdoc in Youssef Idartour's lab at NYU Abu Dhabi. And today, I'd like to share with you some of our work on the impact of ethnicity on the single-cell immune response to malaria in West African children. Malaria is one of the leading causes of child death in Africa, with more than 240 million cases reported in 2021 alone, and more than 600,000 related deaths, with 79% of them being in kids in Africa under the age of 5. And particularly 11 endemic countries carry the majority of the disease burden in Africa, including the Nigeria, the DRC, and Burkina Faso. And so 98% of the total cases are due to infection with the parasite Plasmodium falciparum. And this is typically transmitted by the female Anopheles mosquito, whereby upon infection, it can cause a range of clinical manifestations from mild or asymptomatic to mild and very severe. And while there are different factors that contribute to this clinical severity, the spectrum or the way that our immune cells are involved in the initial encounter and parasitic clearance and building the immunological memory, and also in parasite re-encounter upon re-exposure to the parasite is very important in determining how severe the disease will be. And so as many of us know, the African population is genetically widely diverse with an extensive population substructure. However, it is largely underrepresented in many genomic studies and GWAS studies. But of particular interest was a particular ethnic group that drew the attention of many, and that is the Fulani ethnic group in Africa. That are known to be less susceptible to severe malarial disease, with having lower infection rates, lower parasitic density, and also higher serological protection. And so we leveraged this information in order or in an attempt to answer one of the main questions that we have in mind, which is what are the immune correlates that confer lower disease susceptibility to malaria? And to tackle that question, we teamed up with the CNFRP team in Burkina Faso, where we have recruited 126 children between the age of 5 and 12, both male and female, 58 of them being from the Fulani ethnic group, and 68 being from the St. Mosi ethnic group that live together in the same villages. And here are some of the pictures that can help you appreciate the complexity and the challenges faced with such field work. Essentially, some of them were non-infected, as determined by microscopic detection of parasites in the blood smear, and some were indeed infected. However, they were not presenting with any symptoms. And so, using the blood that was collected, we from the 126 children, we performed in-field peripheral blood mononuclear cell isolation. These PBMCs were frozen and shipped to Abu Dhabi, where we performed pooled single-cell RNA sequencing with a 10x platform, and obtained more than 70,000 single-cell transcriptomes, and performed our downstream analysis with cell type identification, pseudobulk differential gene expression analysis, and EQTL analysis. Just to break down these steps a little bit, these 126 samples were essentially also genotyped. That will help in the pooling and demultiplexing afterwards. So we randomized the samples to prevent any batch effects rising into 21 pools of 6 samples, after which demultiplexing of the sequencing reads and alignment was done. Demultiplexing of the single cells per individual to their corresponding sample was also performed, followed by cell type identification by Seurat, and quality control filters that we have added for more accuracy. And this left us with a total of 71,784 cells from the 126 individuals. And so after that, we performed a reference mapping-based technique in which we have projected the 70,000 and so cells onto this reference UMAP, obtaining around 30 different cell subtypes, as you can see here. And on the UMAPs to the left, that are divided by infection status, infected and non-infected, and classified by ethnic group, you can see a kind of an even distribution of the Fulani and Mosi cells across the different cell types, but not particularly so in the monocyte populations, where you can see kind of a divergence between these two ethnic groups. And so to look further into that, we performed a global principal component analysis, taking into account the pseudobulk transcriptome of all the cell types per individual. And what you can clearly see is a striking clustering based on ethnicity, with red being Fulani and blue being the Mosi. From there, we then performed differential gene expression analysis by Muscat, where we could highlight that the majority of the number of the differentially expressed genes are carried by the CD14 and CD16 monocytes indeed. And a principal component analysis of the cell type specific transcriptomes of these two cell types, you can also see clear clustering based on the two ethnic groups. And so what are the genes that are differentially regulated? So just looking at the hierarchical clustering heat map with the top 25 differentially expressed genes, you can see a clear clustering. This is the Fulani compared to the Mosi in the infected CD14 and CD16 cells. And what I wanted to highlight is the downregulation in the Fulani of many genes in both cell types that are involved in pro-inflammatory responses, including many interleukins or cytokines, as well as chemokines. Some of these pro-inflammatory genes include interleukin-6, TNF, interleukin-18, and interleukin-1 beta, where you can clearly see that there's a lower expression of these genes in the monocytes of the Falani, regardless of infection status, highlighting that there is a general reduced pro-inflammatory gene expression signature there. And another interesting observation that we have seen in the cells of the Falani is that generally across all cell types, there was a particular upregulation of metallothionein genes, genes that are part of the metallothionein family MT2 and MT1, that are specifically upregulated in the Falani across all cell types. And these genes are known to essentially be upregulated in immune cells under stress or upon the availability of metals in the serum, and their role is to have an anti-inflammatory or immune regulatory response there. So just to put all of these genes in a gene set enrichment analysis using the hallmark pathways, it was quite clear to see at this point that there is a general downregulation of inflammatory responses in the monocytes of the Falani. And this goes in line with previous studies in which that show that upon re-exposure, and these children are usually re-exposed to the parasites in these villages. So upon re-exposure, monocytes tend to differentiate into an immune regulatory phenotype to avoid immunopathology and aggravation of the disease and falling into the severe category of malaria. So after that, we were interested in kind of looking into what are the contributing factors for the genetic and lifestyle factors that can contribute to the variation that we're seeing in the transcriptome between the Falani and the Mosi. And for that, we've done a gene by environment expression quantitative trait loci analysis at the cell type specific level. And this analysis was nicely done by one of the undergraduate students in the lab, Jakob. Essentially, from this, the SNPs that were genotyped, around 1 million of them have been put into the model where they have a minor allele frequency of more than 10% per ethnic group, and that they lie within 100 kilobase region of the gene in which they might be associated in regulating. And from there, around 250 SNPs have passed Bonferroni correction. You can see them all highlighted in orange, and this is specifically for CD14 at the top and CD16 at the bottom. And I just want to bring to attention two interesting examples per cell type. And one is a SNP that regulates MT2A, the metallothionein gene expression in CD14 cells. And there we can see kind of a genotype by ethnicity effect, where individuals that carry a major, 2 copies of the major allele are denoted here in 0, and the one denotes for individuals that carry 1 or 2 of the minor alleles. And similarly, another SNP that regulates GBB2 expression, which is an interferon-related gene in CD16 monocytes specifically. And there we can also see this genotype by ethnicity effect. So essentially, here is where I want to wrap up and just leave you with some key messages where using single-cell RNA sequencing, we could show that ethnicity in our population drives the majority of the variation of the transcriptomic data, with the monocytes having the greatest number of differentially expressed genes, and that our differential gene expression analysis of monocytes have shown a general reduced pro-inflammatory signature in the Falani, highlighting an immune regulatory phenotype that could also explain some of the eventual phenotypes of them having lower susceptibility to the disease. And also our QTL analysis has enabled us to identify some of the SNPs that could be associated with the regulation of gene expression, and that could also be further modulated by the lifestyle differences between the two ethnic groups that can contribute to the variation in disease susceptibility. And finally, I just want to leave you with this model for malaria pathogenesis, in which ideally one would aim for having asymptomatic disease with more controlled inflammation and more efficient and effective responses in pathogen clearance and memory building. And this is essentially contributed by several factors, including our genotype and lifestyle, and highlighting these things will be of certain importance to our full understanding of the disease. So with that, I'd like to acknowledge, of course, the children and the families that have contributed to the study in Burkina Faso, the CNFRP team in Burkina that have immensely helped in recruiting and processing the samples, and of course, Youssef Idardoud and the entire team, all of which have been greatly involved in the project. Yeah, thank you. And I'm ready for your questions.
Thank you, Tala. That was a supremely interesting talk, and really nice to see how you were able to take working with the team in Burkina all the way through to the lab.
Thank you very
as
much.
always, any questions for Tala, please drop them in the Q&A. In the interest of time, I'll give everyone another sort of 10 seconds to drop their questions in. Otherwise, please drop them in while we have our next speaker. Okay, thank you, Julia. Tala, I don't know if you can see the question, but the question comes, how do you avoid batch effects with 10x runs or between 10x runs?
Yeah, sorry, I'm not sure why I unmuted myself. What we tried to do is that essentially during the pooling process for the single cell runs, we had infected and non-infected samples, as well as Mosi and Falani samples being pooled together per 10x run that were later on demultiplexed bioinformatically in the end. And also during the process of sampling and recruitment, it was as these children were living in the same villages, the processing of the PBMCs by isolating them and the freezing and the shipment was all done simultaneously and all together in a randomized manner, because at this point we've de-identified the samples. And so this is how we technically sustained or minimized batch effects. I hope this answers your question.
Thank you, Tala. Thank you, Julia. I'd like to now present our final speaker of the session, Dr. Priyam Narain, who is a postdoctoral fellow at NYU Abu Dhabi. Priyam, when you're ready, please share your slides. Oh, and we can see them.
Yep. It's fine. You can hear me as well, right?
Very well.
Yeah. Hello, everybody. I am Priyam Narain, postdoctoral associate in Professor Dipesh Chaudhary's lab at NYU Abu Dhabi. So here I'm going to speak briefly about two projects where we have used single cell sequencing in the mouse brain. So just a brief about like the human brain or the mammalian brain. So the brain itself is very complex and is composed of different types of cells, or which are the neurons, which are the main players. However, besides the neurons, there are the other cells, the non-neuronal cells, which are also very important. Of the non-neuronal cells, the main subtypes are the glial cells, the oligodendrocytes, the microglia, and the astrocytes, of which the astrocytes are involved in very important function of homeostasis, neuronal growth, and neurotransmitter cycling. So why do we study mouse brain, and how can we use the studies from the mouse brain to like scale up to the human brain. So of course, the human brain is much larger than the mouse brain and of course, much more complex. But there are certain levels of similarities as well at the level of the individual neurons and the connections. For example, a study recently found out that there's a circuit, the inhibitory signaling circuit, which is kind of conserved between the human and the mouse brain. So there are similar studies as well, which have found out similarities in the circuitry at the levels of the neurons in the human and the mouse brain, and which is why it gives us an impetus to study mouse models and to use the data from these studies for the human samples. So there are numerous single-cell studies which have been conducted in the mouse brain nuclei. Of some of these are just highlighted here, and this is like important, giving an insights into the the different cell types or the composition of the mouse brain nuclei. Today, I'm going to speak about, the title of my talk said, that circadian rhythms and mood disorders. So what are the circadian rhythms? So there are two types of circadian rhythms. The one is the molecular rhythm and the other is the electrical rhythm. So the molecular rhythm is actually the transcriptional translational feedback loop, which takes place over a course of 24 hours. And the key players in this molecular rhythm are the clock genes, which are the purr, the cry, and the BMAL and the clock. At the electrical level, there is the rhythm where there is the electrical mechanism where in the neurons of the SCN, which I'm going to explain later in my slide, that there is a high firing during the day and a low firing during the night. And this is classical observed both in the humans and in the rodent models as well. So where does all this happen? So this actually happens in a very important nuclei in our brain, the SCN, the suprachiasmatic nucleus. The suprachiasmatic nucleus is the principal or the master clock in our brain. So it receives the input from the retinohypothalamic tract of the eye. So the light from the environment gets into the eye, and via the retinohypothalamic tract, it gets into the SCN. And from the SCN, it is then transmitted through the projections where the SCN is linked to other nuclei in the brain. And that's how we are entrained to the light and the dark, the thing in our, the day, the 12 hours dark and the 12 hours day, which we have. So, just a minute. So there is a link between circadian rhythms and the mood disorders, because it is well known that the perturbations in the circadian rhythms can lead to different disorders, especially the stress, mood regulation, and aversions. And how does the disturbances in circadian rhythm leads to this is because what I call as a broken clock. When there is a disturbance due to shift working or due to irregular sleep or due to jet lag, the outcomes can be seen at the level of disorders which involve depression, the disturbances in the the HPA axis, obesity, and metabolic disorders. So today I'm going to speak, the first project which I'm going to speak about uses a rodent model, the GFP mice model, where the GFP reports the rhythmic expression of a clock gene, which is called the period 1 gene. So classically, as I said, that the SCN neurons have high firing during the day and low firing during the night. However, in a study conducted by Me Nobel in the year 2009, it was found out that there is a subset of this neurons which is marked by the rhythmic expression of GFP, that there is an extremely hyper excited or an extremely depolarized state. However, this depolarized state is not observed in the non-EGFP expressing neurons. So we were excited to study that what is it that leads to this hyperexcited state in the subset of the neurons in the SCN, which classically show high firing during the day and low firing during the night. And why this is important is because this hyperexcited state did not lead to the death of the neurons. It was just that they were silent during this duration of during this period of the day, after which they started firing again. So this was like, what is the molecular mechanism driving this kind of hyperexcited or a depolarized state? We implemented single cell sequencing on the nuclei, the SCN, the suprachiasmatic nucleus, or the master clock of our brain, across 6 time points along the light-dark cycle. So here, this is 3 time points in the light and the 3 time points in the dark, and ZT is the Zeitgeber, and lights on is actually ZT0, and ZT2 is 2 hours after lights on, and 6 is like 6 hours after lights on. So once we had the samples from all these 6 time points from the SCN, what you see here is actually the SCN from a GFP mice, the transgenic mice we have, and these are the GFP neurons there. However, we took out the whole SCN from here, and each of these time points had 2 biological replicates from which the data was integrated, and what you see here is the integrated UMAP from all the time points from the whole SCN of the mice brain, which after clustering and using the classical Seurat pipeline for analysis, we got the 24 clusters of cells here, which were all mixed of neurons and the non-neuronal cells as well. So next, we wanted to identify, so when this study was ongoing, this was the first study and there was no study as such out to help us identify, like in terms of like the single cell studies, which would enable us to annotate these. So we used the classical canonical marker genes, which were published through bulk sequencing data to identify the neuronal and the non-neuronal cells. So here is, as you can see, so we use SAL4 for the neuronal marker and AGT, like the angiotensin, for the astrocytes, the PLP, the PGDFRA for oligodendrocytes, and so on. And that's how we annotated our clusters, and we identified the neuronal clusters, which you see here, and the non-neuronal clusters above here. So while this study was on, and next, since we had like 6 time points and 2 biological replicates from in our samples, we wanted to make sure that we have an equal representation of those clusters from all our data sets. So we also individually analyzed all of our time points, including all the biological replicates, and we could see all the neuronal and the non-neuronal cells were equally represented in all of those time points. So once all of this quality check was done, we wanted to specifically look at the subtypes in the neuronal cells of the SCN. And why? Because this is where the difference between the physiology of the cells was observed in the paper I had just discussed before in the previous slides. So from the neuronal cells, we picked up from the whole cluster, we picked up our neuronal cluster and subclustered it. And this cluster was again annotated on the basis of the known markers. While this was underway, a paper was published which actually did a single cell sequencing on the same nuclei. However, the time points were different. But since the canonical genes remain conserved or like are expressed irrespective of the time of the day effect, there's no time of the day effect on them, we used that genes as well. And what we could find out was that our data from here, the clusters were well matching with the published data. However, we were more interested in looking at that what are the gene expression differences between the neuronal, between the EGFP and the non-EGFP cells. Just to remind, because these are the EGFP cells which are which are extremely depolarized during a time point in the day, whereas the non-neuronal, non-EGFP cells don't show that electrophysiological property. So the clock has the SCN has several neuropeptides, which are like the canonical neuropeptides, and this data is well known from the previously published bulk data. So we also wanted to see at the single cell level or and zoom in more as to which subtypes of the neuronal cells in the SCN express those specific neuropeptides. So what you see here are the AVP, NMS, VIP, VIP, GRP. So all of these are the neuropeptides which are involved in signaling within the SCN. So once we had annotated our clusters on the basis of these neuropeptides, we were also interested in looking at where is the EGFP cell type is expressed from all the clusters. And we could identify, it's not visible here, but we could identify 2 clusters where we could see the expression of the EGFP cell type, which was the VIP-NMS cluster and the AVP-NMS cluster here. And what you see here, the violent plots here actually also show the clock genes. So these clock genes are the part of the transcriptional translational feedback loop. And we wanted to check what are the genes, which clock genes are expressed in the EGFP clusters and which are not. And this helped us identifying the clock and the non-clock clusters as well. Next, we wanted from the single cell data as well, we wanted to look at the rhythmic expression of the clock genes and at the cluster level. So since we had already annotated our clusters, we used the average expression function from the CURAT to plot the average, expression over 24 hours at like time point, or the time points we had 2, 6, 10, 14, and see the time series expression as well. And the other goal was to identify, is there a difference between the expression or the phase of the clock genes in the EGFP versus the non-EGFP cluster? And this helped us in identifying that most of the clock genes are in phase in both the clusters, except for PER1, where we see some differences at ZT2 and at ZT6.
Priyam, just a gentle reminder to wrap up kindly. Thank you.
Yeah, sure. So I will just quickly go through how many minutes do you have?
We're actually at time, I'm sorry to say, but I can give you another 30 seconds.
2 minutes more,
Sure.
and I'll be done. Yeah. So quickly about the next study where we did the single cell sequencing on the nuclei called the habenula, which is actually involved in mood disorders. And through this study, we exposed the mice to a chronic stress paradigm where we get the subtypes, which is resilient to stress, and the other subtype, which is susceptible to stress. And through this study, we were able to identify the clusters, the neuronal and the non-neuronal cells in the habenula, and able to identify which subtypes of the cells are involved in specific depressive or the mood, the phenotypes which we see post the chronic stress paradigm. And from there, we wanted to specifically look at the lateral habenula. So the habenula, which you see here, is composed of the lateral and the medial habenula. And we were specifically interested in looking at the phenotypes which the lateral habenula neurons encode. And from this, we were able to identify that specific subtypes of the lateral habenula neurons are associated with the specific phenotype, what you see here. So just a quick thing about like what is SI, SPT, and TST. So SI is basically it's like a post-chronic social stress. We phenotype the mice and we classify them on the basis of the tail suspension, the sucrose preference, and the social interaction test, because the TST indicates anxiety, SPT is kind of anhedonia, and SI, which is the social interaction, indicates the social avoidance, which are the phenotypes observed in a depressive-like disorder or in mood disorders. So this single cell study enabled us to map the specific cell types involved in the different phenotypes which we see in different mood disorders. So yeah, sorry, I had to wrap up. Just like this is our Chaudhary lab at NYU Abu Dhabi, where we are using a chronic stress model to study the effect of stress on sleep, and also the effect of stress and circadian rhythms. So in the end, I would just like to thank my team members at the Chaudhary Lab, my supervisors and mentors, Professor Dipesh Chaudhary and Professor Justin Blau, and two amazing undergrads who have worked with me on both these projects, Alexa and Marco. Thank you so much.
Thank you, Priyam. What a great presentation. Really interesting findings. We have run out of time for Q&A, but I encourage everyone to join us for the next session, which will be about empowering the Middle East HCA engagement to achieve scientific goals. To join that session, I encourage you to click on the agenda page and join click the Join Now button at the top of the screen. But first, we're going to have a quick 15-minute break. So Fereshteh, Tala, Priyam, really thank you for your talks, and have a great day, everyone.